So go ahead, Dr. Popovich.
Thanks, Kedar. Morning, everybody. And again, welcome to our first Buckeye Neurotrauma lecture. This is, as Kedar just told you, this is a new open seminar series sponsored by Ohio State's Chronic Brain Injury Program and also the Center for Brain and Spinal Cord Repair. And this morning, I'm honored to be able to introduce to you our inaugural speaker, Dr. Jonathan Godbout. Dr. Godbout completed his PhD and his postdoc training at the University of Illinois. As a postdoc, he was awarded an F-32 fellowship from the NIH to study mechanisms underlying the effects of neuroinflammation and aging. After his successful postdoc, which is when Dr. Godbout established himself as a bona fide leader in the field. We were fortunate enough to be able to recruit him here at Ohio State. He's been here about 15, 16 years now. And during that time, he's risen through the ranks, first in the Department of Molecular Virology, Immunology, and Medical Genetics, which is where I used to be a faculty member and when I first got to know and interact with John. And then more recently in the Department of Neuroscience, where he is currently a tenured professor. Dr. Godbout has also or continues to serve in various leadership roles in the college, the university, and in the research community. Specific notable roles include his service as Assistant Director of Basic Research in the Institute for Behavioral Medicine Research, where he's been a primary member for a little over a decade now. As co-director of the Neuroscience graduate program, managing editor for Brain, Behavior, and Immunity. And since 2018, he's been the faculty lead for Ohio State's Chronic Brain Injury Program. On the research side, Dr. Godbout runs a very large and very well-funded laboratory and has mentored some of the most impressive PhD students that I've had the pleasure of interacting with. He publishes often and in some of the best neuroscience journals in the world. To date, he has 90 career publications, and the collective impact of these papers have helped establish Dr. Godbout as a leader in the field of microglial biology, especially in the context of understanding how this resident CNS myeloid cell can affect the brain's response to stress, aging, and traumatic brain injury. So I could go on and on about John's accomplishments and his contributions to the department, the university, and to the neuroimmunology research community at large. But I think you'd rather hear him speak than continue to listen to me. So I'll end by saying it's my great pleasure to welcome Dr. Godbout as our first BNL lecturer. And today he will talk to us about his recent work characterizing the role played by trauma-associated microglia in promoting chronic cortical inflammation, neuronal dysfunction, and cognitive decline. Welcome, John, and we look forward to your presentation.
Yeah, thank you, Phil, and thank you, Kedar, for introducing me. And certainly my pleasure this morning to talk a little bit about our research and, tell you about some of our recent data. And as Phil said, I'm part of the neuroscience department and chronic brain injury, and you can follow me on Twitter or check me out on my webpage if you want to know more about me. I think I have to give you disclosures. I don't have any disclosures that would be confounding. Just the work here is supported by 2 NIH grants, an R56 and an R01 grant. So today I'm going to tell you about our traumatic brain injury project, and I just wanted to start by giving you a little bit of background. And I think it's important to talk about how many people are affected by traumatic brain injury, from the very young to the very old, and a lot of times these injuries occur by falls. We see a lot of traumatic injuries with accidents. Many of you are on college campuses, including OSU, and we have these crazy scooters, and every place that has these scooters, an increase in head injuries. And of course, there's a lot of press about sports-related injury, whether it's the NFL, NHL, or youth sports like soccer and hockey, lacrosse. We know TBI affects our military personnel with penetrating injury, but also a lot of concussive injury from blasts. And one thing we've learned a lot more about in recent years is this domestic violence associated head injury, and actually seeing quite a bit more of this during the COVID quarantine. So a very serious situation in that area. So it affects a lot of different people, from the very young to the very old, in a lot of different ways that you can become injured. And I like this iceberg analogy. I know other people have used it, and so I took it from them. But a lot about what we see with TBI is on the surface initially. You come in and you could be unconscious or have brain swelling or inflammation. But a lot of it actually is sort of what bubbles under the surface that's dangerous. And I think we see that with the high degree of neuropsychiatric problems that occur with TBI. I mean, one thing that was striking to me when I first started TBI research was the 10 times increase in depression people have. And this risk doesn't go away with time. It seems to increase. There's been very few long-term studies on this, but the ones that have done it seem to show an increased risk of depression after injury that really never goes away, increases with time. We also see functional recovery problems, sleep disruptions, anger management issues, concentrations. One thing I think we hear a lot about is this cognitive decline and potential risk for increase in neurodegenerative disease, which is a serious issue. And these things are sort of bubbling again underneath the surface like we're seeing with this iceberg. And there's also life and social impacts where people with brain injury tend to turn to alcohol and drugs of abuse more. Some have trouble holding a job. Some have trouble with managing finances. Suffer from loneliness. So it's really a compound problem. And I think the one stat I think is important to take away is that over 70% of all people who have a traumatic brain injury continue to have secondary neurocognitive and behavioral complications. So that's a striking number of people that continue to have these issues under the surface. And that's why I think this iceberg analogy is really good. So as Phil mentioned, I'm really interested in inflammatory processes in the brain. And are these inflammatory processes the underlying cause of some of these neuropsychiatric problems that people develop with time? And if so, what cells are really responsible for this? And if you look at the cells of the brain and spinal cord, I think a few culprits come up as being main targets. And that's why so many people are interested in these particular cells. And so for us and many others in the field, we concentrate on the resident innate immune cells of the brain and spinal cord, the microglia. Okay, about 10% of your CNS population are microglia. These are resident tissue macrophages of the brain and spinal cord. We also have CNS macrophages that come from the periphery and sort of are just visiting in certain areas of the brain and spinal cord, aren't permanent residents, but still can contribute to long-term inflammation and pathology. And we also have the contribution of peripheral immune cells that often influence the course of injuries. But these are really probably the targets of a lot of people's research, both clinically and basic. Okay, so a little bit more about the microglia. They're these macrophage-like cells. They're surveying the microenvironment. They're an important immune sensor. They're involved in structure of the blood-brain barrier. We know that in development, they're important for synaptic pruning and brain development, as well as through the course of lifespan, monitoring synapses, interacting with astrocytes and neurons. And I think when we think about their activation state, we're talking about with infection or injury, we see more of an activated profile, often leads to structural changes that you can see by histology. This is a more active, deramified microglia, which can make chemokines, cytokines, reactive mediators, complement components, a lot of things that you'd associate with that innate immunity. And this is tightly regulated. So you want to be able to control this level of inflammation. Our brain, spinal cord can tolerate some level of inflammation, but you want to be able to shut it off. So they're tightly regulated by other microglia, astrocytes, potentially neurons, and maybe other immune cells like monocytes and T cells. So one thing that we've been interested in the context of aging and stress and injury is this idea of priming or sensitization or reactivity, a lot of different words for it. But let me just walk you through this a little bit, what I'm trying to say about priming. So with injury, we know there's going to be a certain level of inflammation, right? That's not going to surprise anybody who's within this virtual conference seminar. We know with activation, we see that these microglia change shape. We know that they produce things like inflammatory cytokines like IL-1, TNF-alpha, chemokines like CCL2 and CCL5. And these are potent activators of the immune system, also help recruit cells to the site of injury. And again, this is tightly controlled. And with time, you usually see some resolution of this inflammation. But there is some that goes unresolved. So there's a level, a profile, a number of cells, maybe probably region dependent, that you don't see that resolution. Okay, and one of the profiles we see is primed in that it has a lot of innate immune markers still increase. It's not necessarily active. It's not making cytokines or chemokines anymore, but it has markers associated with antigen presentation, even some dendritic markers like CD11C, CD14, CD68, which are innate immune toll-like receptor or scavenger receptor things. So it's more of a kind of a heightened state of reactivity. And we also know that some of these cells probably become dysfunctional and aren't doing what they're supposed to be doing anymore, not clearing debris, not acting as an important immune sensor, maybe dysregulated in some way. So there's certain populations of these cells that persist, I think, both in the rodent models of injury and in humans of this primed and dysfunctional. If you want to learn a little bit more about priming, a grad student in my lab, Christina Witcher, wrote a nice Trends in Neuroscience paper on that. And we've done a lot of work with this. And sort of one of the consequences of this prime profile, and we've looked 30 days or 1 month after injury, is that in this heightened state of inflammatory sensing that we're seeing with the microglia, is that if you give an immune challenge, so we've done peripheral immune challenges with lipopolysaccharide, you see amplified and exaggerated microglia activation within the brain to this peripheral challenge. And this results in cognitive impairment, depressive-like behavior, kind of reactivation of a lot of the inflammatory pathways. So it seems to exaggerate what's happening and leave the animal in a sensitized state for weeks to months after injury, which we think is really a relevant area of research and work that was done by Ashley Fenn and Megan Microglial in my lab. Now, in humans, they also seem to see this other, reactive or inflammatory profile of microglia or macrophages in the brain. And this is work done by, the first slide here is Jennifer Coughlin. These are in former NFL players. They had relatively a short history in the league, 3 to 5 years, were relatively young. She's looking for a PET imaging of benzodiazepine receptor. And what you see here is this brighter color showing you more binding. It's unclear if this is really microglia activation or some change in activation state, but certainly people with a history of concussions here have higher levels of this. She's not the only one to show it. There's a study in the Annals of Neurology showing, again, these are different patients with a single TBI at different ages showing some of this increased PET ligand binding of this benzodiazepine receptor. So certainly in human studies of imaging and postmortem studies, there is evidence of changes in microglia and macrophages long after the injury. So I think this is, and there's a lot of data obviously I'm not showing you, but I think what we're seeing is that the microglia component is important driver of inflammation. And we wanted to look at two things really: how microglia change over time, and what is their influence on other cells. And that's going to be the majority of what I'm going to show you for the lecture. And so what I want to kind of walk you through is some of the data that we've shown and others about these phases of TBI and the profiles of microglia. And starting at the acute phase, this is going to be the initial injury. You're going to see inflammation, obviously, after head injury. You're going to see changes in the blood-brain barrier. You're going to see swelling or edema. You're going to see the peripheral immune system responding. You're going to see intracranial pressure. You're going to see different profiles of microglia where you see the highly activated ones. You can see phagocytic microglia. You see macrophages within that site. And the idea is that these things are there designed to help clean up debris, help with starting repair processes. And in general, these are thought to be important biological processes that are protective. Now, if you move a little bit past this acute phase, you go into the subacute phase, which in mice is a week or so after injury. In humans, it's going to be a little bit longer. And you see this secondary injury processing happening. So you're going to see some ischemia, glutamate toxicity, and inflammation. And if we look in the mice, what we see is a unique set of profiles. And here the microglia in the subacute phase still tend to have some inflammatory capacity. We see these really interesting rod microglia, and these show up particularly in the cortex, where they form these long trains of microglia. This is a picture from Christina's study showing a kind of a train of these rod-shaped microglia. We're not exactly sure what they do, and we've really tried to understand them, but they're certainly present. They're present after TBI, and they're actually present in a lot of neurodegenerative conditions as well. And then with the chronic phase, so in mice, this is about a month later or longer, we see evidence of cell dysfunction, some levels of inflammation, and potentially even neurodegeneration. And we don't see, really active profiles of microglia. What I think you're seeing is this increased readiness to respond, this prime state, and potentially this dysfunctional state that is no longer doing what it's supposed to be doing. So I think it's pretty clear there are distinct phases of microglia, and it depends on the time frame and probably the region that you're looking at. So what the model we've been using is this midline fluid percussion injury. And in 2010 or so, I know that I went to Phil and said I wanted to do more work within this space, and could he help me? And he did, and he set me up with a great researcher, Dr. Johnny Lifshitz, who was at Kentucky at the time and now at the University of Arizona. But we learned from him, and he's helped us in a lot of our studies since then, and been a great collaborator. So what this model is a midline craniectomy where you're attaching an injury hub and a connection here to this fluid percussion device, and it provides a fluid pulse that impacts the dura, and it makes the brain move like this. So it's sort of down and out. And so what we see here is that in this injury, we get this nice diffuse injury with injury to the cortex, the hippocampus, the thalamus, and actually the cortex here splays out and hits the skull. So you see a lot of interesting changes within the cortex that are due to the mechanical injury and the rotation forces, and also just the inflammatory responses that are going there. And the reason that we like this method is that it gives more of a diffuse injury, very similar to what you'd see with a coup counter coup movement within concussive injury, like I'm showing here with the football helmet. And you see widespread neuronal injury or evidence of neuronal injury and inflammation. And so this is a study that Christina did, starting to look at the time frame of these responses. We initially use these nanostring encounter gene arrays. We use the mouse version 2, which has 250 genes plus some of the genes of interest for us. And we took the cortex specifically because it was an area where we saw a lot of interesting microglia changes. We saw the rods, we saw the reactive profiles, we saw the activated, and she compared the acute time period at 8 hours and the subacute of 7 days. And these data are published in Glia, so I won't go through a lot of them. But I think just to kind of show you the transition over time is that you see a lot of inflammatory genes initially. So within that 8 hour to 24 hour period, and that's here in the left here. And you see things like IL-1 beta, IL-6, not surprising. And then things that are sort of show up in both at 8 hours and 7 days. You see a lot of chemokines, things associated with toll responses and interferons. This Venn diagram kind of showing you that there is 17 uniquely regulated genes, both at 8 hours and 7 days. And then at 7 days, again, more 33 genes here, a lot of them associated with tolls. Some of our markers of priming show up, CD68, MHC2, a lot of interferons, parts, and tolls. So there seems to be, and not surprisingly, a transition from that acute phase to that subacute phase. And the thing that really struck out to me was the transition associated with interferons. So this is some results. The nice thing about nanostring, these large panels, is there's a lot of genes, and you can run some of these higher order upstream regulator analyses. And so what kind of pops out is that initially just to highlight here is that in that early 8-hour period, you see a lot of NF-kappa B mediated genes and MAP kinase. And again, not surprising, you're driving a pretty strong inflammatory response within the brain. But at that 7-day, you start to see a transition towards interferon type 1 signaling, which is fascinating because type 1 interferons are mostly respond to infection, so a viral infection. So why is this response being highly driven at 7 days after injury? And what Christina did here in this paper, it's published in Glia, this is just the title. She found some interesting changes in microglia. She also eliminated microglia and saw the effects on these profiles without microglia present, and did a lot of work trying to understand the presence of rod microglia in the cortex. But she built on this initial project and wanted to do more time points and more genes. And we switched to a nanostring neuropath panel, which has a lot less inflammatory genes, but a lot more genes associated with neuropathology. So genes that would be in astrocytes or neurons or oligodendrocytes. And here she used the colony signaling factor receptor antagonist. Probably everybody has heard of this, a very useful model system in neuroimmunology where you can feed this compound to mice, and within about 7 days, you get a complete or 95% depletion of all the microglia in the brain and spinal cord. Also seems like it may affect some of the other long-lived tissue macrophages, not to the same degree, but to some level, and doesn't really affect the bone marrow or circulating levels of monocytes. So what you did here is, I should say that we removed microglia prior to injury. So that's one caveat of the study. So there's no microglia when the mice are being injured. So we do that 14 days prior to the injury. They get the control sham surgery or the brain injury, the midline fluid percussion. Then we're going to compare 1 day, 7 days, and 30 days, which represents our acute phase, our subacute phase, and our chronic phase. And again, we're going to focus on the cortex, and we're going to run these different genes. And this is just showing you kind of the time frame of what's happening, just to highlight again that there's no microglia in the brain when we're injuring these mice, at least in a fourth of the mice, and then, or half the mice. And then these different profiles in the control, we'll see the activated, we'll see the sort of intermediate, we'll see the rods at 7 days, we'll see the primed, and then at 30 days, I think we see some primed and dysfunctional microglia. This is just showing you what the results of Plexicon look like when you put the mice on the diet. And remember that we put it on before injury and we kept them on the entire time. So there's no microglia. This is IBA1 labeling in the cortex. You see these IBA1, these are little microglia here. You can't see them super well, but they kind of look like this. And with the Plexicon, there's a 95% reduction based on histology. So this I think shows, there's a lot of data with these, and I'm going to just try to highlight some of these things. And this paper was recently accepted in Journal Neuroscience, and we did the proofs this week, so it should be out soon if people want to take a look at that. There's a ton of data and a ton of data that's been submitted to the online registries, as well as a bunch of supplemental data. So if you guys are data nerds like myself, then you'll have lots of fun looking at that, and we certainly did here. So this is a result of the nanostring analysis of the 750 genes and looking at the upstream regulator analysis, so the canonical pathways and upstream regulators. And you can see here these heat maps show you sort of the darker orange here are activation or increases, and the blue is decreases. And we see canonical pathways associated with neuroinflammation evident at 1 day, 7 days, and 30 days after injury. Not surprising based on a lot of the work that we've done. But for most other things, you see that it sees a much stronger response here at 1 day, and that at 7 days, we see that these pathways are somewhat activated. And by 30 days, not much evidence of things that remain, just maybe the neuroinflammation, the TNF-alpha pathway. A lot of these other things kind of go back to being restored. Now, if we eliminate microglia prior to injury and we do the same time course, you see that there's really ablation of all these things. So not only is there not a response initially after injury in terms of these inflammatory pathways and upstream regulators, we see that persists, that you basically ablate inflammation early and it really never is there at any time point. So in my mind, this helps solidify that really these microglia are really the main drivers of persistent inflammation. And here's another way to look at it, looking at all these data. This is the acute time point, again, from the cortex. This is what I'm trying to show you here in the pie chart is the percentage of genes. So 114 genes were differentially regulated at 1 day after injury in the cortex. And 67% of them were microglia independent, meaning that they were still increased or decreased independent of microglia elimination. So only 33% of these genes were microglia dependent 1 day after injury. And the genes that were increased by TBI and reversed by the Plexcon treatment were things like MyD88, CD14, interferon response element 7, TNF alpha, so things that we know are driven to some degree by the microglia. And what you see is over time is that more of it shifts. So there's 61 genes that are still elevated in the subacute phase in the cortex at 7 days, and 70% of them are microglia dependent. So now the number that are microglia independent is going down. So you're seeing that a lot of things that are still being driven at 7 days are microglia dependent. And by 30 days, there's even more, about 80% of them. And again, the genes that show up here in the cortex at 30 days after really fall into that priming profile of CD11C, MHC class 2, CD68, CD14. So things we know are associated with that more reactive profile of microglia, which is totally ablated if you eliminate the microglia. So we became very interested in the different cell populations and the effect of microglia on other cell types. So we did single cell sequencing, which I'm sure everyone's familiar with now. We needed help with this. This is not an area of our expertise. So we reached out to Dr. Andy Fisher, who's at OSU, who had been kind of really showing some cool data at our neuroscience faculty meetings. And so we're excited to get him involved in this and do some of these studies using 10x genomics, where basically you make single cell suspensions from the cortex. You pair them up with these gel emulsion beads to get a single cell, which is sort of packaged with the primers and to uniquely tag these transcripts. And then you use the RNA for sequencing. Here's a result of that from our study from the cortex. And we focused here again on that subacute time phase. We did it with or without microglia at 7 days. And we focused on the cortex. So these are the different representations of the cells. You see that we see microglia, oligodendrocytes, astrocytes, endothelial cells, neurons, and go through the whole thing. And there's different subpopulations of each of these cell types. One nice thing about single cell sequencing, at least at 10x genomics, you get really nice representation of microglia and astrocytes, less so of neurons, and there might be some ways to improve that by doing different techniques, but this is what we started with, and so these are the data that I'll show you. We'll focus here on the microglia. You can see them as TMEM119 positive, and some of the neurons profiles, which are MEG3 positive. So this is the different clustering of the microglia. There are 10 distinct clusters. Now, one thing I'll tell you about clustering is it's a little bit subjective, sort of like when you first learn how to do flow cytometry and you realize that you can subjectively move the gates. It's also true with the distinct clustering. So, I mean, if you wanted to have 45 clusters of microglia, you can. If you wanted to have 2 clusters of microglia, you can. So it's a little bit of an art form, I think, to get the most representation of clusters based on the gene expression. So Christina Witcher did this work, and I think she did a good job of trying to balance that and came up with 10 distinct clusters. And we see here on the right is how these clusters fall out based on the treatment groups. So again, we have the vehicle control, the vehicle TBI, the Plexcon control, and the Plexcon TBI. So the Plexcon TBI, there's a lot less microglia, but you can start to see some unique changes with the different treatment groups. And so when I talk about single cell sequencing, I try to break it down in a way that makes sense to me. And I have a very small caveman brain. So the best way for me to talk about it and show you is to use pie charts. And I think they work quite well. So let me go through the profiles of the microglia in the cortex 7 days after injury by the single cell sequencing. So the most representative populations I think we see here are in the controls, are the population cluster 3 and cluster 4. So cluster 3 has a lot of these RNA transcripts that are associated with resting or surveying or homeostatic microglia. And there's been a lot of single cell papers published, so you can look and confirm that. And this population 0 is quite strong. And then 7 days after injury, you see that the percentages shift. So we see an increase in a lot of populations, which pop out as cluster 8, cluster 2, cluster 9, and cluster 6. Probably the ones that really caught our attention, I think, are cluster 6 and cluster 8. And I'll go through and explain a little bit, why I think that's true. But cluster 8 and 6 have a lot of interferon-related genes. They have some inflammatory genes, chemokines, things that we've seen before. This cluster 9 has a lot of that disease-associated microglia, something that some really high-profile papers have come out and seen in ALS and MS and Alzheimer's disease. We see a similar one with TBI here. And cluster 2 looks like a population that might be damaged or potentially dying with a lot of ribosomal proteins. So there weren't a lot of cells left over with Plexicon, but there's some. And so we want to look at those profiles too. And I just want to show you those. Again, here's what we're seeing with TBI. We're seeing a reduction in the homeostatic genes clusters of cluster 3 and cluster 4. And a relative increase in cluster 6, 8, 9, and 2. And I think the pie charts show it pretty well. In the Plexicon alone group, you see that really an increase in cluster 2 and 7. 7 is unique to this population. And with the Plexicon TBI, we see changes as well. But the ones that really are prominent are increases in cluster 7 and cluster 2. So cluster 7 turns out to have a lot of genes associated with growth and survival, and cluster 2 has a lot of things associated with dysfunction and death. And so what I think is happening is that when you're Plexicon, as long as they're on the Plexicon drug, the microglia are kept in a either kind of cycling state where they're constantly being killed and then regenerated. So you get I think that's what you're seeing here is a constant turnover of those microglia, because once you take the microglia off the drug or take the mice off the drug, the microglia all come back within 3 days. So there's a strong promotion of the growth here in the context of also balance of their destruction. And I think we're capturing that a little bit. So this is a cool way also to show the data of some of the top hits of the genes associated with each cluster. The little circles show you the relative amount of cells that are expressing these genes, and the number, and then the color darkness shows you that the relative amount of expression. So a couple to kind of highlight here, this cluster 6, which I told you had a lot of interferon related genes, and the reduction in homeostatic related genes like TGF beta receptor, the prunergic receptor Y12, the fractalkine receptor, and TMEM119. So as you see some of these markers increase, we see the homeostatic genes decrease. And this makes sense that if you want a microglia to change and be more activated, you have to reduce a lot of the things that are involved in their regulation. We've seen this before multiple times in different models that we've done. This cluster 9, again, is also the one that kind of shows up as disease associated. Again, reductions in homeostatic genes, but increases in things like ApoE, CD52, TREM2, and CLEX7A. And a lot of people think that this cluster 9 is actually might be involved in repair. And we see this, at least some evidence of this happening, because this cluster 9 is also enriched in the Plexicon TBI mice. Now we can look at the analyses of these upstream regulators here, and we see a number of things going down here at 7 days, but a lot of things going up. And all the things in red, again, are tied to the type 1 interferon response with interferon alpha, interferon response factor 3, STAT1, interferon alpha receptor, alpha 1, and beta and alpha, and even a type 2 response with interferon gamma. So again, more really concrete evidence supporting our data with the nanostring. So just to sort of summarize some of these data, this is my interpretation and what we used in the paper and what we've talked about at lab meetings. Clusters 3 and 4 seem to be the surveying microglia, the homeostatic microglia based on profile. Cluster 7, which only shows up in the plexicon control groups, looks to be a repopulating group. The cluster 2, which shows up with plexicon and TBI, seems to be a damaged or depopulating cell type. And then we see these cluster 6 and cluster 8, which I like to call TRAMs, the trauma-associated microglia, which is sort of the title of my talk. These guys seem to be the ones that are really elevated with the injury and seem to be the ones that are very strong in interferon signaling. And this cluster 9, I think I've talked about. And then those of you interested in some of the rod microglia story that we've pursued over the years, but one of the reasons that we did single cell was to see if we could figure out which profile was the rods, and I don't think we did. But based on the transcriptome and the data that we could put together, they fall somewhere in between this cluster 8 and cluster 9. And we actually think now that these rod microglia may actually be important for repair, and they may not actually be cells that are negative to driving inflammation, but might be positive impact. And I know Johnny Lifshitz just recently published a paper more on the profile of these rods. So they're of interest. We just don't know exactly what they're doing still. Okay, so how do microglia influence over time? I think that we see diverse profiles at acute, subacute, and chronic time points after TBI. And I think the microglia are absolutely critical in those persistent inflammation and reactivity that we see at these subacute and chronic time points. So I've been hinting at this as we've gone through, but this relevance to interferons is fascinating to me with my kind of immunology and biochemistry background in grad school and as a postdoc, because what interferons do is really important antiviral responses. And this is in the context of a head injury, right? So why do we see such a strong interferon response in the microglia after head injury? And one thing I can tell you going back to immunology is that if you want to prime a macrophage, what you do is you give it interferons. So that increases their readiness. We see that, oops, sorry, I didn't mean to go forward. You can see that they're associated with antigen processing, antigen presentation, debris clearance, phagocytosis. So all the things that we would associate with priming. So we may have uncovered, what is the main cause of priming 30 days after injury? Likely is this interferon pathway, and we're looking at that more closely. And we're not the only ones either. There's a nice paper here from David Lone's group in collaboration with Alan Faden from University of Maryland. David now is at Trinity College in Ireland, but they show some nice data showing interferon beta, which is a type 1 interferon. It has a detrimental role in inflammation and neurodegeneration in the controlled cortical impact model of TBI in mice. So certainly supporting evidence that these interferons are important. And the pathway that kind of stuck out to me in terms of trying to understand what interferons might be doing and how cells are responding to them is this CGAS-STING pathway, which STING stands for the stimulator of interferon genes. You find that it's kind of a potent transcription factor that drives type 1 interferon genes, and it senses damaged cells. So we know with TBI, particularly in the cortex, there's going to be a lot of mechanical damage. We're going to see changes, neuronal damage, damaged mitochondria. I know a lot of people in TBI are interested in changes in mitochondria. So STING helps sense this and could be, what they're picking up here. And we don't know this for certain, but the genes it helps drive are the IRF3, which we see prominently in the nanostring and the single cell, and driving these type 1 interferons. So Chelsea Bray and Jonathan Packer. Chelsea is in the MedPath program, and Packer is a neuroscience graduate student who just started. As you can tell, he's very excited about the project. But we're asking this question, what happens to inflammation, pathology, and functional recovery after TBI if we knock out the STING pathway. So if we block the sensing of this response and driving interferon genes, what happens? And so him and Chelsea have wild type and STING knockout mice. We gave the same control and TBI that I've shown you before, and we're focusing on those same time points I've highlighted at the subacute 7 days and the 30-day chronic. And so far we've started to analyze these data. We have a lot more of it. I'll just show you 2 highlights. This is microglia at 7 days after injury. They're Percol enriched. So what you're doing is increasing the number of microglia that are in that population by using density Percol gradients. It's about, you get 70, 80% pure microglia, which is better than just doing cortical samples. And here we're looking at a number of those interferon genes. The idea here is if you knock out a key driver of interferon genes, you should see a reduction. So this is what they're seeing here. This is one of the interferon response genes. It's increased like we've seen before at 7 days after injury. And if you don't have the STING knockout, global knockout for right now, this is reduced or back to baseline. Same is true in the interferon response factor 3, which is one of the things that STING really drives. You see that here at 7 days after injury, it's increased in microglia. If they don't have STING, that's back to baseline. And same with 68, another gene that we think is being driven by this pathway that appears in microglia. 7 days after injury is increased by TBI and then reversed by the STING knockout. So I think that's really good, because if you knock out a key driver of the interferon pathway, you're hopeful that the interferon genes will be affected, and they are. And looking at some of the behavior, one of the things that we've seen pretty reliably 30 days after injury is that there's some cognitive impairment. I think it involves both the hippocampus and the cortex, and to sort of capture that, we've done novel object recognition and location. And what we've seen is that in the recognition test, you basically give them 2 objects, and then in the third trial here, you give them a new object, and the idea is that they should spend the most time with the novel object. We see that in the control groups, whether they have STING or not. In the TBI groups, this is significantly reduced, and with the STING knockouts, this is sort of intermediate, so an improvement. Same is true with location. What you do is you change, you have 2 different objects, and then you just change the location of one of the objects. And again, we've seen this main effect of TBI that seems to be attenuated by the STING knockout. So we're very hopeful that we're on to an important pathway in that transition from the subacute to chronic phase. And we have a lot of things going on to help prove that. Okay, so how do microglia influence other cell types? What I'll show you today is neurons, and I think this is pretty fascinating. This again is looking at the single cell. We found, or I didn't find, Christina found 11 distinct clusters of neurons here. Neurons here, they're not as many neurons as you'd hope to, and maybe one way to improve that would be to just do single nuclear sequencing. But still, we have a pretty good representation of both excitatory and inhibitory neurons based on these different RNA markers here, whether it's CAM2 or Thi1 or GAD. You see or different types of neuron pools. And what we found here is 7 days after injury, the majority of genes in neurons were suppressed by TBI. So if we look at these genes and we look at them in the context, again, remember we were doing this with or without microglia. So we can look at what happens with TBI and then TBI if the mice don't have the microglia. And so some genes were increased, and the genes that were increased with TBI are really glutamate related. Not surprising, because there is glutamate toxicity as part of the secondary damage profile with diffuse injury. Those are increased, but then most everything else associated with homeostasis and plasticity or decreased. And you kind of see that here in the upstream regulator analyses, where you see things associated with growth and survival, like NRF1, IGF1, HIF1 alpha, other pathways here that are, increased or decreased by TBI. So it seems to have TBI has a pretty profound effect on the neuronal profile at 7 days after injury. So that the pathways that were increased overall in these gene expressions from the neurons were synaptic long-term depression, Glutamate receptor signaling is increased, but most everything else was decreased, like calcium signaling, dopamine signaling, synaptic long-term potentiation, endocannabinoid signaling, and synaptogenesis. So really seems to show you that 7 days after injury, you see some changes in neuronal homeostasis and health. And I think the really fascinating part of this study is that when you look at the effect of microglia depletion, we found at 7 days, there were 233 total genes that were differentially expressed, okay? And the majority of them are reversed. So all the changes happening in the neurons, if you eliminated the microglia, have gone back to normal. And that's shown here in the blue. So 193 of these genes were reversed. So a lot of these changes that we're seeing are gone. Partial reversal is just, it means it's attenuated, it's moving towards full reversal, but not quite. Only 10 of these genes, only 10 of the 233 were unaffected. So it seems like a lot of the changes that are happening 7 days in the neurons are microglia dependent. Okay, so gene transcription is one thing, but are we actually seeing functional differences is the next question. And so this is the same setup. Here, this time we use these Thy1 YFP mice, and the reason that we used them is because you can see the GFP reporter, you can see it in the neuronal soma, in the apical dendrite, and within the axon. So you can visualize these. About 10% of the neurons light up in this version of the Thy1 YFP mice. So we did again what we did before. We eliminated microglia prior to injury. We focused on the 7-day time point, and we focused on the cortex. And we did some, we looked at some shoal analysis and dendritic complexity. This was actually done by a really amazing exchange student from Thailand, Titikorn Konchaya, who, I just butchered his name, but I called him T-Korn. So he did some really nice tracing here, and I think you can see pretty well that with the control versus the TBI, that structurally the neurons in the cortex look a lot different. And we see sort of a reversal if you eliminate microglia prior to injury. And probably the best one to focus on here is these cortical shoal intersections. So looking at complexity, when we see a main effect of TBI here at 7 days, that's reversed by microglia elimination. So some differences in complexity that seem to be microglia dependent. We also looked at dendritic remodeling. We looked here again using the YFP mice and looked at the number of dendritic spines. Now, there wasn't a difference in the number of spines. What T-Korn found was there was a difference in the type of spine. And so we see at 7 days after injury, a modest reduction here in the number of mushroom spines, which are thought to be mature spines, and an increase in stubby spines, which are thought to be immature spines. And we don't see these same effects in the mice where you've eliminated the microglia. So again, more evidence that there seems to be plasticity issues 7 days after injury that likely are microglia dependent. All these things sort of we come together with this idea of neuronal connectivity. And we know that a lot of these cortical axons project across the corpus callosum. And so we got help from Dr. Candace Asquith at OSU in the ePhysiology core, where we looked the same experimental design that I've showed you. We looked 7 and 30 days later looking for changes in neuronal connectivity using some ePhysiology. There's a lot of data here, and I'll just show you kind of the highlights here. I'll focus on this. The main difference was the amplitude of the N1 versus N2 signal, which is supposed to show you the myelinated versus the kind of unmyelinated fibers within the corpus callosum. And what we saw was a main effect of TBI on the ratio. Here you see it at 7 days, so a reduction in the N2 to N1 ratio. And we still see that here at 30 days in the TBI group, but this is restored back to the baseline if you've eliminated microglia prior. So there seemed to be some long-term benefit of eliminating microglia on the context of neuronal connectivity through the corpus callosum. And finally, Christina and Chelsea looked at cognition. Again, we use the novel object location and recognition. which should tap into both cortical and hippocampal type memories. The first one we did was the novel object location, and we found, a deficit with TBI at the 30-day time point, and time spent investigating the novel location. And this discrimination index, which I showed you before, shows you that the mice, generally prefer the new location, whether they're control or Plexcon TBI, but the TBI mice don't. So they don't really localize to the new object. We also did a number of rotorod and horizontal bar studies to look at movement. And what we see is that there's really no deficits in these type behaviors at 30 days. What we do see is some cognitive differences. And this same sort of track follows through with the recognition. Again, something I've showed you before, looking at the changing the object to a new object. And the idea is the mouse is going to want to investigate the new object, which we see here. 80% of the time in the controls, even the Plexcon alone. Remember, they have no microglia, but their memory is just fine. If any of you guys have that type of question, and then we see a TBI dependent reduction in cognition that's restored by microglial elimination. And this discrimination index is just another way to look at the percentages. And again, you see that there's really no preference for the TBI for the novel object, and that's restored if you've eliminated microglia. Okay, so many of you probably, maybe you've stopped listening to me, or you don't care, and you say taking away microglia prior to injury is kind of crappy and is not very relevant. And I would agree with you some degree, but not fully. I think it gave us a lot of interesting data and put us on to that interferon pathway, which I think is tremendously important in trying to understand the transition between the subacute to the chronic inflammatory phase. But we know what you're talking about, and we thought, another way to approach this is to use this depletion repopulation strategy, or what is also called forced turnover. And so the reason you can do this is because as long as the mice are on Plexcon diet, and you saw this with the single cell sequencing, that they kind of are in this constant state of cycling, but basically they remain eliminated as long as they're on the Plexcon diet. You don't see very many microglia. But as soon as you take them off, within a day to 3 days, the microglia start to come back, right? So you can do this depop repop study or forced turnover, like many have done in the past. And this is work that people have actually done in TBI as well. Some nice work again by the Faden Lone group, as well as Mark Rutenberg and Jana Vuvik, who are Australian, show this benefit of doing this, taking away microglia and then letting them repopulate, and then being involved in neural repair and recovery. And we've used this forced turnover protocol similar in models of stress with Mike Weber and Shane O'Neill in a model of aging. In aging, we weren't very successful at turning over some of the profiles. They kind of remained inflammatory. But we wanted to take this approach here, and I think we were smart about our decision in terms of the timing. So we wanted TBI to occur, and we wanted the initial inflammatory reaction to happen, because I think a lot of argument can be made that initial acute phase response is important for recovery. So we waited 7 days after injury at sort of the height of that inflammatory or interferon subacute phase. So we put the mice on the diet at 7 days, and then we went to 14 days and then stopped the diet, and the microglia repopulated for 16 days. And so this is just showing you the IBA1 labeling from the control group. You see nice little green happy microglia. When they're depleted, they're gone, so you don't see any. And then at 30 days after injury or 16 days of repopulation, you see that the microglia are back. And this is from the TBI group. We again did the cortex. We did it a little differently. We didn't do the microdissection. We just did gross cortical dissection. We did the nanostring neuropath panel again, and we looked at some cognition. This is work really done by Chelsea Bray in the lab. And I think the results are quite striking. So this is a heat map of the gene changes at 30 days after injury. So this is cortical genes 30 days after injury. And we see here is, I think, a total of 53 genes increased or decreased by TBI. and you see that the vast majority of these are reversed or attenuated if you've done the microglia repopulation. So again, we're seeing those differences in inflammation pop up at 30 days that are totally reversed if we recycle or turn over the microglia. And there are some main effects of repopulation by itself that probably can't be discounted. So we're unsure, if microglia come back exactly the same as they were prior to being depopulated, but for the most part, it seems like they do. There were a few differences here. I think you can see them up here where there's kind of a main effect of repopulation. But overall, I think if you look at an interaction between TBI and repopulation, we see a reversal of the majority of genes. And again, a lot of the genes here that are going down are associated with neurons here. And I'll show you a little bit of that. So again, this is looking at the effect of the interaction between TBI and the microglia turnover intervention. And we pop these data into Ben Barris's cell-specific type of database where you put in the gene, it tells you what CNS cell likely expresses it. So we had 53 genes expressed 30 days after injury in these cortical samples, and we found that 48 of them were microglia dependent in that the turnover reversed their effect, and only 5 were unaffected. So the ones that were affected here, I think the ones that really stand out to me 30 days is the effect on neurons. So a lot of these genes are going down, things that maybe some of you guys in spinal cord injury are interested in, like NOGO is being suppressed, neurotension, this neurofilament, which I know people use as a biomarker in neurodegeneration, which is actually increased here at 30 days. All these, this is the glutamate receptor, all these things are reversed by the microglia turnover. So there's a benefit to turning over the microglia on the neuronal homeostasis 30 days after injury. So we also looked at some behavior at 30 days. The reliable behaviors we've seen that persist or kind of develop 30 days after injury, there's 2. There's the cognition, which I've shown you before. There's also some evidence of increased depressive-like behavior or increased resignation. This is done in a tail suspension test 30 days after injury. And we can see here is the only group that kind of has immobility in this test 30 days after injury is the TBI group. And this is attenuated if you force the turnover of the microglia. And then the same is true looking at the cognition. Again, it's been a very reliable test for us in the lab, where we always see deficits 30 days after injury in the novel object location, which I've shown you several times now, and also shows up in that discrimination index. And again, the repopulation was quite beneficial to this and restored that cognitive function. And same is true with the novel object recognition, where there's a TBI effect and a reversal with the repopulation. So this is work that Chelsea Bray has done in the lab, and she's actively working on getting this paper out, and hopefully will be submitted shortly. It has a lot of more data than I'm talking about, but these are some of the highlights I wanted to show you. So with that, and probably my time here, I want to give you some conclusions, some of which you've already heard, but microglia had some diverse and unique profiles that were evident at the acute, subacute, and chronic time points. I think understanding these drivers of the profile, including type 1 interferons, is very important, and to me represent key targets of intervention because they're involved in that transition from subacute to chronic inflammation. So I think if you're a clinician out there, I think that is something that I would like to talk to you guys about for sure, because I think that is the future of some of the stuff that I would like to do. I know that microglia are directly responsible for that persistent inflammation after diffuse TBI, at least in mice. And I know that the ablation of microglia in neuroinflammation prevented all the differences in neuronal homeostasis, plasticity, and cognitive issues. And if we can even target, microglia during that phase, during that 7 days, and take them out and let them repopulate, and they come back as a new cell, that was very beneficial in lowering that inflammatory profile and improving cognition, and also improving some of the markers of plasticity that I didn't show you. So with that and my time, I'd like to thank the lab. Unfortunately, we haven't had any good pictures taken within the past years. So these pictures are a little bit older, but we've had a good group. We do fun stuff, and it'll be exciting to get back to seeing them every day again and doing fun things like Christmas parties and bowling events and other things. But the people in my lab that really drove this, former trainees Ashley Fenn and Megan Mucci-Grosso were instrumental. Current people in my lab, including Christina, Chelsea, Shane, Zoe Packer, and several undergrads have been part of this project, and we wouldn't be able to go anywhere without them. They've done a tremendous amount of work. We also have a new grant that will be starting soon to continue with this work on TBI and interferon signaling. Then I've had a lot of great TBI collaborators at OSU, many of you probably on the call, along with some outside investigators like Rachel Rowe, Johnny Lifshitz, Audrey Lafreniere, and Rowan Walker. So with that, I will end, and I welcome any discussion or questions that you may have.
Thanks, John. We had a couple questions crop up here. So for anybody who's out there, you can use the Q&A tool to ask your questions. You can also raise your hand and we'll call on you as we get to them. The first question that we received was, for nanostring, was it done on the cortex tissue or the microglia isolated from the cortex?
The nanostring was all cortical tissue.
Okay. And then I did see a hand from, I think it was Dana McTighe. If she still has the question, raise your hand. Otherwise, for anybody else, you're welcome to ask your questions now.
I have a comment rather than a question, Kedar.
Sure, go ahead, Phil.
So, John, great talk. It was really fascinating to see a lot of, I haven't seen a lot of that data, so congratulations. Just a comment. So one of the things in spinal cord injury that we see when we use the exact same PLX diet is really an exacerbation of pathology and outcome in the absence of microglia. And I think it has a lot to do with the scar forming phenotype that you see at the lesion site. We lose a lot of that, and it exacerbates inflammation and changes a lot of the pathology and functional recovery profiles in those animals. So I'm thinking in many cases in the clinic, people who suffer spinal cord injuries also present with various severities of traumatic brain injury. So based on your data and the data that I'm aware of in our lab and other labs in spinal cord injury, a systemic microglial suppressing drug may not be ideal, because it's going to have, I mean, based on the data that I've seen today, it may have conflicting effects in the brain versus the spinal cord. So any idea how we can develop or approach a therapeutic from a targeted region specific or brains or versus spinal cord specific manner?
Yeah, I think you bring up a great point, and certainly that is something that people have seen in a couple different models. I think it might depend a lot on the contribution of peripheral macrophages, because in diffuse injury, in the brain injury, we don't see a very big or really not a significant contribution of peripheral monocytes and macrophages to the injury site, what I think you do in spinal cord injury. And likely that Plexicon does not affect the bone marrow production of monocytes, so you're still going to see a pretty good level of monocytes in injury potentially. So I think there's a balance there. I also think that timing is important, and I think that there's some models of TBI as well that if you take away microglia prior to injury, it's detrimental. So I think that's why the forced turnover during the chronic phase or potentially the subacute phase is what's important and what we're moving towards. And I think in that case, it might always be beneficial to do it that way. The other thing would be to use some of the cooler new nanotechnology, nanoparticle targeting. I know some people in CBI have dabbled in this, looking at new ways to package inhibitors and put them directly in the tissue of interest. So maybe there'd be a new technology, but I think, like you said, I mean, inhibiting any pathway globally can be negative.
Thanks, John.
Andrea Tedeschi has a question here. Said, great talk. How are changes in the environment, maybe day 14 after TBI, would impact on microglia repopulation? So how do changes in the environment maybe after day 14, would impact on microglia repopulation?
Andrea, I don't know the answer to that question. I know they repopulate very quickly, and they repopulate independent of whether the mouse was injured or not. I mean, certainly by 1 day after injury, you see some level sorry, 1 day after removing the diet, you see some repopulation. So they come back pretty quickly. I don't know, like if you're talking about environmental enrichment, would that enhance it? It might, but they seem to come back, and they seem to come back, uh, a lot similar to what they did when they left. Unless they were injured, then they come back more in a naive profile, which is what we were trying to get out of it. But I don't know the answer to your question.
Okay, and Jill Ward says, thanks for the talk. For your rod-like microglia story, have you considered using digital spatial profiling, also made by the NanoString company? Are the rod-like microglia lining capillaries or vasculature or axons?
Yeah, they actually line. So that's part of the glia paper that Christina published, and they are aligning against the apical dendrites of the damaged neurons. So it seems to be very specific about where they're aligning. And those neurons don't die. So that was one of the thoughts where we think that they're involved in sort of the survival of neurons over time. And we actually see that population enriched in the Plexcon TBI group. So I think it's very likely that maybe one of their roles is repair. And I do think that slide seek or those other things that NanoString has would be great, and definitely something that we want to do. I think Phil and I were discussing that right before we went live. So absolutely, if that's an expertise of yours, let me know, and we'd be happy to learn from you.
All right, thanks, John. Benjamin Brett says, from a clinical perspective, do you suspect that pre-existing inflammatory or viral conditions could influence timing or intensity of microglia response post-injury?
Absolutely. Yeah. So I think so. When you think about, and let's start with the more easy way to explain it. Let's say you have a head injury, okay, and you do have this more sensitized CNS state, and it probably affects several populations, microglia, astrocytes, neurons potentially, and so not all of us get secondary head injury, right? And so if you look at all the findings and saying repeated head injury, we see worse decline in neurodegeneration, but most people don't get multiple head injuries. So the 70% of people that still have neuropsychiatric problems have only had one head injury. Most of them are not severe. So 94% of those people didn't have a severe injury. So what is it in their life that sort of exacerbates these problems? And so all of us get exposed to viruses, as we're seeing with coronavirus, how fast and how large that footprint is about us being exposed to pathogens. So the immune system in the brain communicates. So every one of those challenges, even if it's peripheral, will relate to the brain. And the brain responds by changing reorganizing the host priorities, changing behavior. So those we think are really strong, prominent drivers of change within the brain. So it could be a peripheral infection. It could be other stressors. Nikki Kokaiko-Cochran's doing sleep disturbances. We also know that psychological stress could be a major one that drives some of these things. So absolutely. I mean, a good example for you is to look at what happens in Alzheimer's patients with infection. And so if you look at the cognitive decline of an Alzheimer's patient, it's not a linear curve down. It's a spiked going up and down. If you were to look at when it goes down, it's often with another inflammatory event like an infection. So absolutely, I think infections can be drivers of cognitive and behavioral issues associated with TBI.
All right, thanks, John. Another question here. Have you tried to spatially map the microglia clusters using in situ hybridization ISH techniques like RNA-scope? Maybe you're able to identify the rod microglia.
That's a great comment, and we are doing that as we speak. We are working on, we're not experts in RNA-scope, but we're developing it. Two people in my lab are working on that idea. Becca Biltz and Red Yin are looking at RNA-scope, and we hope to be able to do some of that as well. That's a great suggestion.
All right, thanks, John. I think that is all the questions we have, and we're a little over our time. So we may just conclude here. So thanks very much to Dr. Godbout for presenting today, for Dr. Popovich's partnership and introduction this morning as well. We'll be back in a few months with actually a presentation from Dr. Popovich, so stay tuned. You can always learn more about what we're doing with this series on our Twitter page, at Ohio State CBI, as well as our website, discovery.osu.edu/CBI. So thanks very much, everybody, and have a good rest of the morning and good weekend.
Thank you, everybody.